I'm Paola, and I work in this building. My laboratory is literally sitting on top of this auditorium. And as you can guess from my accent, I'm from Italy. And I've been working in 4 different countries before coming into this big global institute. And today, I would like to tell you about a much longer journey. Yes, and I would like to start from here. This first slide reminds us that the future has its roots in the past. And as Stanley Kubrick in 1968, in his masterpiece, 2001: Space Odyssey, anticipated a trip of mankind through space to the very nature of humanity. From Greek mythology, we have learned of Prometheus, punished by Zeus for having donated fire to men. The punishment was that an eagle each day would eat his liver, that during the night would have grown it back, would have regenerated. So what do we know now? Is that liver is one of those organs that can regenerate. And how many other organs can regenerate? And if we want to look at the very future, we'll be able one day to rebuild, even in the laboratory, an entire organ. And nowadays, we know that organ indeed regenerates thanks to the presence of stem cells. You might have heard about stem cells before, and I would like just to give you this simple slide to explain what they are. A stem cells is a specialized cells, well, is a spatial cells, I would say, that is able to give rise to cells that are of 2 types. One is identical to itself, so the new stem cells, and the other daughter cells will give rise to many different types of tissue, becoming specialized tissue of our body. And I would like you to keep in mind that each of our organs regenerate at a different pace. We change all our gut epithelium in 4 days, all our skin in 3 weeks, all our muscles about 3 times in all our life. The branch of medicine that studies organ regenerations, organ and tissues regeneration, is called regenerative medicines. And regenerative medicine uses stem cells instead of the classic drugs in order to repair tissues. It also uses vectors, they are modified viruses, to correct genes in our body. And nowadays, it also utilizes biomaterials. And in modern times, the first cell therapy has been the blood transfusion at the beginning of the 20th century. And this allowed to save millions of lives, especially during the First World War. In the Second World War, with the advent of the atomic era and its deadly radiation effect, we had this fact stimulated the development of bone marrow transplantation, which was the first stem cell therapy. And this has been done with a great contribution of Donald Thomas, who won the Nobel Prize for this. And this therapy now saves more than 1 million lives. And since the bone marrow transplantation represents still the paradigm of all the regenerative therapies nowadays, I would like to explain a bit how it works. A patient who has a disease in their blood, because bone marrow gives rise to all our blood cells, gets ablated of the stem cells in the bone marrow. Nowadays, we don't do anymore with radiation, but we do it with drugs. And the patients receive a bone marrow that contains stem cells from a healthy donor. So then the healthy stem cells engrafted in the body will give rise to a healthy blood and save patients' life of many, nowadays, of many types of disease, including tumors and like leukemia. More recently, in the early 90s, researchers here at the Great Ormond Street Hospital, at the Necker Hospital in Paris, at the San Raffaele Hospital in Milan, developed a novel therapy that allowed to treat these diseases, which are congenital immunodeficiencies. These kids are born essentially with no immune system because they have a corrupted gene. And they would not be able to defend themselves to any infectious agents that we daily fight against with no problems. And the researcher at the time took the bone marrow of these kids and transduced, put, let's say, a corrective version of the genes into the blood stem cells, and then put back in the same patient their own bone marrow, this time corrected, with stem cells healthy. And these kids now, there are more than 30 which are treated and became young, healthy adults. Before this therapy was available, these kids were not living long, and they were actually living literally in a bubble. Stem cells can also be grown in a dish, and the pioneer of stem cells expanded in a culture dish is Howard Green, professor at Harvard and mentor of many important and famous stem cell biologists. And what he did is being able to develop a culture system for which you can take a small piece of skin, maybe for a patient that went through an injury like a large burn, and the stem cells grown in the lab could be able to cover up to 80 or 90% of the entire body. And one of his students, who was my PhD actually advisor, discovered that a single skin stem cells would have the potential to cover large part of the surface of this big room. And of course, these stem cells would not disclose this potency under normal conditions. But if you challenge them in the proper way, they would do it. And if you take these clones and grow them in a dish, then you can recreate an epidermis. Then piece by piece, you can replace over an injured body up to the total surface of our body. And this now is a life-saving treatment for thousands of hundreds of people. This helps me to introduce a much more recent story, and the story of Hassan. Hassan now is a kid, happy to play football with his friends. However, 3 years ago, he was completely skinless. He's a Syrian refugee who arrived to Germany. And the moment he arrived, he lost 80% of his skin, as he represented in the drawing there. Michele De Luca and another students of our Green from Italy flew to Germany, took a little piece of the spare skin of Hassan, brought it back to Italy, grown in the laboratory. And this type, he had to do something more. He had to correct the gene, because Hassan represents the first butterfly kid being cured ever. These kids have a genetic, they have a genetic disease for which their epidermis detached from the below tissues. So these kids are afraid to touch anything. They cannot play soccer. They live their life completely covered by medicated dressing. And what Michele did was to expand enough cells to cover 80% of Hassan's body by correcting each cell with the normal gene inside. And I would say that only 20 years ago, this would have looks like science fiction. And now, in the last few minutes, I would like to tell you how many laboratories in the world, including ours, are trying to think how to repair other tissue, other organs, maybe inside our body, that might have different challenges. And the next few minutes, I would like to tell you how we are planning to replace an esophagus. If you think to rebuild an organ, you should start from, a way would be to restart from their shape. And one way to get an organ shape would be to take a real organ from a donor and strip out all the cells. And now we can do this in the laboratory through infusion of detergents and various solutions. Once you get this structure, this scaffold, which we call natural scaffold, we could combine it with expanded stem cells in the culture dish and ideally to be able to put it back into a patient. What we have done recently is taking an esophagus, and in the first row you see that it contains cells. Those blue dots means that they are nuclei of the cells. While in the lower row, you see how the organ remains empty of cells, no blue dots, but the scaffold, the structure of the organ is maintained. Then we have re-put back the cultivated cells of different layers of the esophagus. And we have done, for example, how it's shown in this video. Of course, this is, I'm showing with the color solution, but just to give you an idea how we can control, by pressure, how to locate the cell suspension and leave it in the right place where we want. This is actually how it looks like, the esophagus, inside this structure that is called bioreactor, because we can maintain the organ pretty alive, and we can also stimulate it with subject to activity that would be like inside the body. And in the lab, a bioreactor actually looks like more like that. And when we take it out after a certain period, the organ has reacquired quite a few of the original features. So why, and you might have heard this has been reported by the BBC, and as the title says, is that this was done on a small scale, on a small animal size. What we now want to achieve is to rebuild, to scale this up to an organ for a human size, and especially to children's size. Children born, for example, with this atresia, so the esophagus does not develop properly, and the current treatment is pulling up the stomach into the thorax. And this, of course, doesn't allow the kids to feed properly and have a normal life. That's why, in the future, we hope and we aim to put back an artificial esophagus and put it back in the kids. And I would like just to leave with the very last message, is that the kids represent a great target for regenerative medicines for two reasons. First of all, they are small. If we think to rebuild an organ in the lab, this is an important message for us. However, they have a greater regenerative capacity than most of us. And finally, using natural scaffold will allow them to repair once in their own life, because this natural scaffold will integrate into the body and will grow with the baby. And in the future, we aim to grow different type of organs. Remember only that each of the tissue and organ will have their own challenges. And in the end, I would like just to say thanks, and thanks also to the institution where I work. Thanks.